Good morning, everyone. Today topic is blood. Blood. Study of blood is called hematology. Hematology. Blood is a fluid, connective tissue. Not only blood, lymph also fluid, connective tissue. Blood is a false fluid, connective tissue. False fluid, connective tissue. Because of extracellular matrix of blood, that is plasma. Plasma is not made by any blood cells. Plasma is not made by any blood cells, and they do not have fibers. Hence they are false fluid connective tissue. If not, to become a connective tissue, the following components must and should be present. So what are those components to become here? To become a connective tissue, so the following components are must be present. There this tissue must be present with cell, and also fibers. The cells must be secrete fibers, and the cells must be secrete matrix. So these are the three important components should be present to become a connective tissues. But in case of blood, we have only matrix plasma, but this plasma must and should be produce any blood cells. But here in blood it doesn't happen, and this blood doesn't contain or do not have any fibers. Hence this blood is a false fluid connective tissue. Now coming to the pH of blood. pH of blood is 7.4, and human blood approximately has 5 liters. Approximately it has 5 liters. So this is of blood interactions. Now we will discuss about blood plasma. Color is pale yellow in color. Liquid in blood. So composition of blood plasma. Composition of blood plasma. Generally, blood plasma contain 90 to 92%. 90 to 92% blood plasma contain water. And 8 to 10% of blood plasma contain different solute, which different solute dissolve in water of plasma. So these solutes are like, first one is plasma protein, plasma protein. So in plasma protein, we have most important plasma protein. They are albumin, and second one is globulin. So here albumin important role is which maintain osmotic balance. Osmotic balance, which maintain osmotic balance. Now coming to second one is globulin. So generally in globulin, there are three types of globulins. Proteins are there like alpha and beta globulin, and also very important gamma globulin. So this alpha and beta globulin act as transporters or inhibitors. They act as transporters and inhibitors of, they are mainly helps in transport and inhibition of, and they carry lipids and fat soluble vitamins like A, D, E, K, and they also transport or carry the hormones. So these functions performed by the alpha and beta globulin. Now coming to very important gamma globulin. So this gamma globulin is also called immunoglobulin, are also called antibodies. Antibodies. So we have antibody Y-shaped structure like this. Okay. And these antibodies helps in the are immunoglobulin. They are helps in fight and destroy the they fight and they destroy the disease causing microorganism, are also called pathogens like bacteria and virus. So thus which helps in defense mechanism. So this gamma globulin helps in defense mechanism and provide immunity to the human body and provide immunity. So this is all about the plasma protein like albumin and globulin. So important point here, albumin maintain osmotic balance, and it is the most abundant plasma protein in plasma. So it is the most abundant here, most abundant protein in blood or plasma. So this is all about the plasma protein, so which is present in blood plasma. Now second one, very important, in blood plasma there are different solute like minerals, or also we can use the term electrolytes. So there are various minerals and electrolytes are present like sodium, potassium, and calcium, bicarbonate ion, and chloride, like that. Various minerals are also present in blood plasma, and there are various nutrients present in blood plasma like glucose glucose and fatty acids and acids amino acids. So these are the various nutrients present in blood plasma. Now coming to, in blood plasma, dissolved respiratory gases also present. Dissolved respiratory gases. The dissolved respiratory gases like oxygen and carbon dioxide. And in blood plasma also there is a presence of nitrogenous waste material. Nitrogenous waste material also present in blood plasma like ammonia. It is a highly toxic waste material, and urea, less toxic waste material, and also uric acids. So ammonia, urea, uric acids. So these all are nitrogenous waste material present in blood plasma. So along with this, very important, one more solute or composition of blood plasma is presence of blood clotting factors. Blood clotting factors. So this blood clotting factor also present in blood plasma. Also present in blood plasma. So here two important blood clotting factors are present. So they are fibrinogen, fibrinogen and prothrombin. So this fibrinogen and prothrombin together helps in blood clotting process. Blood clotting process. And a very important note here. Here plasma. Here I am writing plasma minus blood clotting factor. Blood clotting factor is equal to serum. It means that from plasma, removal of blood clotting is nothing but the fluid part of the plasma is called serum. So serum, in simple terms, we can use serum is a plasma without blood clotting. Serum is a plasma without blood clotting factor. Without blood clotting factors is called serum. If the serum contained with blood clotting factor means then it is pure plasma. And if you remove blood clotting factor from plasma, the liquid part of the blood is called serum. So this is all about the blood plasma related important things. Now we will discuss most important blood cells. So in this video lecture we will focus only on RBC cells, and next coming video we will discuss remaining two blood cells. Red blood cells. The number of RBC is differ in men 5 to 5.5 millions. In female is 4 to 5 millions. The shape of RBC during immature RBC shape is oval shape and very important nucleated. It contains nuclei. But in mature stage the RBC is by, and very important here, E. The formation of RBC is called erythro. So this formation occurred during embryo. RBC formation take place in yolk sac, liver, spleen, and also in thymus. So these are the various places where the erythropoiesis process will occur during embryonic stage. After birth or during adult, this erythropoiesis process will occur in red bone marrow. So this is formation of RBC is called erythropigments in RBC. So RBC of most mammals including human do not have nucleus and any cell organelles. So most of the mammals including human RBC do not have nucleus or any cell organelles like endoplasmic reticulum or Golgi apparatus or like mito. All these are cell organelles are absent most of the mammals including human being also. Why? To give minimum space or which provide minimum space to the pigment. The pigment is hemo. Here due to absence of any cell organelles like especially mitochondria, so this RBC perform anaerobic respiration because of absence of mitochondria. So once RBC perform anaerobic respiration, from one molecule of glucose, it gives rise to only 2 molecule of ATP. But if any cells which perform aerobic respiration means, and from one molecule of glucose, it gives rise to 38 ATP or 36 net ATP. But only in RBC, due to absence of mitochondria, it gives rise to only 2 ATPs. And due to presence of this hemoglobin pigment, the color of the blood is red color. Now, structure of hemoglobin is a quaternary proteinous structure. It is made up of with two parts. One is protein part and non-part. Here, protein part is also called apoenzyme part, and non-protein part is called cofactor. This apoenzyme part of the hemoglobin is globulin. A non-protein or cofactor part is called heme is made up of a mineral metals, so that is iron. So iron is a metal ion in cofactor. Now coming to the globulin is a protein part, or also called apoenzyme part. So this globulin, which is made up of with four amino acid chain. In that, two alpha chain and two beta chain. Each alpha chain, which is made up of with 141 amino acids. Each beta chain, which is made up of with 146 amino acids. If there are two alpha chain, means 141 into 2, so equal to 282 amino acids are made by the alpha chain of globulin protein. And here each beta chain made up of with 146 amino acid, hence there are two beta chains, so 2 into 146 is equal to 292 amino acids made by the beta chain of protein part, that is globulin. If you combine these two, then there are total 570 amino acids. 574 amino acids are present, which is made up of with 574 amino acids. And here non-protein part is called cofactor, in which heme is a non-protein part and which is made up of with the iron. So this is about the structure of hemoglobin. Function of hemoglobin. Generally, main role of hemoglobin is it carry R, which transport gases which is dissolved in blood plasma, like it transport oxygen and also transport the carbon dioxide. So if oxygen bind with hemoglobin, it form oxyhemoglobin. In this oxyhemoglobin form only, this hemoglobin transport the oxygen to the tissue, and carbon dioxide bind with hemoglobin, the form of, and then it form the carb amino hemo. So in this form only, hemoglobin transport carbon dioxide from tissue to the lungs. Every 100 ml of oxygenated blood, hemoglobin deliver 5 ml of oxygen to the tissue. Every 100 ml of the blood, hemoglobin deliver 4 ml of carbon dioxide to the lungs. So like that, every 100 ml of oxygenated blood or deoxygenated blood, it is delivered by the hemoglobin to a tissue or to the lungs. So this is all about the structure of hemo. And generally, the normal range of hemoglobin in human is 12 to 16 grams per 100 ml. If less than 5 gram per 100 ml, then it leads to a disease called anemia. Lifespan of RBC generally 120 days or 4 months. So after completion of its life cycle, it undergoes lysis, means destroy. 120 days, RBC goes into two places. Their primary place or primary site is spleen, and secondary place or secondary site is liver. First primary, after completion of its life cycle, RBC goes into two places. Primary place is spleen, secondary place or secondary site is liver. So in spleen, destroy RBC by a process in so hence the spleen is known as graveyard of RBC. And secondary place or liver, so in liver, this Cooper cell destroy the RBC by phago process. After destruction of RBC, from which bile pigment will be released. Bile pigments, so those bile pigments are like bile and bile. So bilirubin yellowish in color and biliverdin is greenish in color, greenish color. Generally, these bilirubins are more carnivorous animals, and this biliverdin more in herbivorous animals. So bilirubin and biliverdins are the bile pigments which form by the destruction of RBC or hemoglobin after complete of its life cycle in liver. So this is how RBC get destroyed in two places like spleen and liver. Now coming to one more important, the hormones essential for synthesis of RBC. The hormone is called erythro hormone. So these erythropoietin hormones are secreted from JG cells here, juxtaglomerular cell of efferent arteriole in nephron or kidney. Hence, erythropoietic hormone is the hormone essential for the production of RBC. Factors essential for synthesis of RBC. So those factors are mainly, some are mostly three vitamins. They are essential for synthesis of RBC. In that, B6 vitamin called pyridoxine and B9 vitamin folic acid and B12, very important vitamins which is essential for synthesis of RBC. So that is Seneco reservoir of RBC. Generally there are two reservoir of RBC. So primary and the larger reservoir of RBC is liver. So liver is the primary or larger reservoir of RBC, and second most is spleen. It is the second largest reservoir of the RBC. So see here, the destroy of RBC taking place in spleen and liver, and blood means blood that has been taken from one person means donor and give to another persons like like recipient within 2 months only, within 2 months or 60 days period. So such type of blood is called transfused blood. Now coming to very very important here, abnormalities of RBC. Abnormality condition of RBC. Here one is polycythemia. Polycythemia. Second one is erythrocytopenia. So here polycythemia means increased number of RBC, which increase number of RBC, that is more than 10 millions. And in erythrocytopenia means decrease number of RBC and hemoglobin also. Hemoglobin also means less than 2.5 millions. This erythrocytopenia leads to anemia. So in which anemia, hemoglobin amount decrease, that is less than 5 gram. Generally normal hemoglobin is 12 to 16 gram, but in anemia condition the hemoglobin amount drastically decrease less than 5 gram. So there are different types of anemias. Generally three types of anemias are frequently we seen in persons. Three types of anemia. First one is microcytic anemia. Second one, megablastic anemia. Most dangerous anemia, that is pernicious anemia. So microcytic anemia is due to deficiency of iron deficiency. Very, very important here. It is the iron deficiency. Due to iron deficiency, it leads to microcytic anemia. Due to vitamin deficiency, vitamin like 3 vitamins deficiency, it leads to cause megablastic anemia. There B6 vitamin deficiency, also called pyridoxine vitamin deficiency, and B9 vitamin, folic acid vitamin deficiency, and B12 cyanocobalamin vitamin deficiency. So these three types of vitamin deficiency which leads to cause megaplastic anemia. Now coming to pernicious anemia, it is a very dangerous anemia, and cause this anemia cause due to deficiency of only one vitamin, that is B12 vitamin deficiency. B12 vitamin deficiency. B12 vitamin deficiency leads to cause pernicious anemia. So this is all about the abnormality condition of RBC. So with this we completed each and every concept related to the RBC. But there are two important topics which is related to the RBC. Now we will discuss those two important topics. So there are two important topics which is related to RBC. One is ABO blood group system, and second one we will discuss RH incompatibility. RH incompatibility. So these two concepts are related to the RBC. Now we will discuss each concept in detail. Yes, about ABO blood group system. So this ABO blood group system was discovered by Carl Landsteiner. Discovered by Carl Landsteiner, and he was considered as a father of blood group. Father of blood group. Example for ABO blood group. Example for ABO blood group systems are complete dominance and codominance, and multiple allelism. So we will discuss all these examples in detail in genetics. And these ABO blood group systems generally present on ninth chromosome, very important. So this ABO blood group system present on ninth chromosome. So on ninth chromosome we have one gene. The gene is called I gene. This gene has three alleles. Generally every gene has two alternative forms. They are called alleles. But this I gene has three alleles. Hence this concept is called multiple allelism. So we will discuss in detail in genetics topics. Those three alleles are here: IA, IB, IO. And generally here this ABO blood group systems, like ABO blood group system contain four types of blood group. Blood group. So these four types of blood group is mainly based on presence or absence of antigen on surface of surface of plasma membrane of RBC. Generally, RBC has plasma membrane. On the surface of this plasma membrane of RBC contain some special type of oligosaccharide structures. They are called antigens, like this. Antigens. So antigens are generally made up with oligosaccharide structures, oligosaccharide sugar chain. If the antigens are present on surface of plasma membrane, then we have different types of blood group. If not only that, but also presence or absence of antibodies in blood plasma. Blood plasma. So finally, four types of blood groups are based on presence or absence of antigens present on the surface of plasma membrane of RBC and presence or absence of antibodies in blood plasma. We have four different types of blood group. Like here we have four types of blood. We will discuss this concept later. Here we have four type of blood group. One is A blood group, B blood group, AB blood group, O blood group. Here A blood group in which antigen A is present. A antigen is present in plasma membrane and B antibody is present in blood plasma. Such blood group is called A blood group. In B blood group, instead of A antigen, here if this is RBC, it is a plasma membrane of RBC, and this plasma membrane contains antigen like this. So this is B antigen. If B antigen present on the surface of plasma membrane, such blood group is called B blood group. So it contains B antigen. In blood plasma contain A antibody. A antibody. In AB blood group, if here, if RBC plasma membrane contain both A antigen and B antigen. A antigen and B antigen. Such blood group is called AB antigen, AB blood group. AB blood group. So in which A and B antigens are present, but no antibodies are found in blood plasma. Now coming to O blood group. In O blood group, no antigens are present on plasma membrane of RBC. So here no antigen, but blood plasma contain both antibodies, antibody A and antibody B. So this type of, based on presence or absence of antigens and antibodies, we have four different types of blood group. Now here, important point here, note universal donor blood group. Universal donor blood group is O positive. We will discuss what is positive or what is negative. Here, universal recipients blood group is AB positive. So this is all about the ABO blood group system. So along with ABO blood group system, there is another blood group is discovered. So that is RH factor. RH factor are also called D factor, or it contains antigens called H antigen. So this factor was discovered by two scientists. Discovered by two scientists. They are Carl Landsteiner, Carl Landsteiner, and one more scientist, Alexander S. Wiener. Yes, Wiener. Wiener and Carl Landsteiner. These two scientists together in 1940 they discovered this RH factor in a monkey. So your monkey name is rhesus monkey. They found in this type of RS factor on RBC in rhesus monkey. That's why the name came as a RH factor. So this based on this factor, based on based on presence or absence of this surface protein, surface protein, there are two types of RH factor: RH positive factor, RH negative factor. 80% of the human population has RH positive factor. Just 20% of the human population has RH negative factors. So these RH factor which determine the blood group whether it is positive or negative. For example, if say this is RBC, so on RBC we have antigen A, and also we have one more antigen, that is H antigen, antigen A and H antigen. If these two antigens are present on the surface of plasma membrane of RBC, this type of blood group is called A positive blood group. And one more important, if RBC has antigen A and also has antigen B and also has antigen H, then this type of blood is called AB positive blood group. It means that on the surface of RBC we have three types of antigens: antigen A, antigen B, and antigen H. Hence it is called AB positive blood group. For example, here universal donor. In universal donor, in an RBC, no antigen A, antigen B. Only there is a presence of H antigen. Only one H antigen only is present. Such type of blood group is called O positive blood group. If H antigen is also absent, then it become O negative blood group. So this is all about the blood group system, ABO blood group system and RH blood group system. Now we will very important concept here we will discuss. So that is RH incompatibility. RH incompatibility. RH RH incompatibility. In which mother has RH negative and father carry RH positive blood group. Their first child fetus. If fetus carries Rh positive birth, then the first delivery gets safe. First delivery become safe because before mother start producing antibody, because before mother start producing antibody against Rh antigen, Rh antigen, baby get delivered. Baby get delivered. If these two persons say carry second birth, again fetus has Rh positive blood group, then the second baby developed Rh antibody in mother. So already if fetus Rh antigen second birth, then already mother developed Rh antibody in during first pregnancy. Here, if second birth is fetus Rh positive, then mother already developed Rh antibody due to first pregnancy, and then it killed the Rh positive baby of second pregnancy, and this leads to this kind of killing of baby. It leads to HDN here nothing but hemolytic disease of newborn. Hemolytic disease of newborn, are also called erythroblastosis fetalis. Erythroblastosis fetalis. So here second birth is Rh positive baby. So their mother already developed Rh antibody against the first due to first pregnancy. So hence the Rh antibody which kill the fetus anti-Rh antigen. So then it leads to a disease called hemolytic disease of newborn, or also called erythroblastosis fetalis. So here first baby get safe, but the second baby get killed if the fetus or second baby has Rh positive. So there is a one treatment for this erythroblastosis fetalis, that is the mother should administrated by the anti-Rh antibody. So before deliver the just before deliver the first baby just before deliver the first baby, the mother should administrate the anti-Rh antibody injections. So then we can prevent the further killing of this fetus by this process, that is Rh incompatibility concepts. So this is on comment box. So here you go with MCQ 1. First MCQs. Yeah. Consider the following statement regarding the blood plasma. So here there are four options are there. So read those four options and how many of the above statements are correct. Second MCQ. Yeah. Yes. Read the following statement about erythrocytes means RBC. So here also four option is there. 1, 2, 3, 4. So read these four options and which of the statement given above are correct. Consider the following statement regarding ABO blood group. Here also four options, multi-statement questions. And which of the statement given above are correct? And one more MCQs I have given.